Okay, this lab is on differential media, so we're going to do two labs. One of them is called McConkey agar, and the other one is called EMB, eosin methylene blue agar media. Okay, and so we have our bacteria here. We have 11 bacteria. We're going to skip number 2 because it won't grow. It's a Clostridium, and it will only grow in areas that do not have oxygen. So we have to put it in a separate chamber. So we're not going to do that one here. What I have now is a split plate. And so these markings are, there's 4 dashes here, and then I have a chart that I'll show you. 4 markings is McConkey agar, 3 dashes are EMB. So there are 2 of these in here, and so I'm going to just show you what it looks like. And it's a little bit lighter for the EMB. So the EMB is the 3 marks. Okay. So you can tell it from a couple of ways. So, but when I make these up, I run a pen, Sharpie pen up the whole stack of them like this, and then I go like that. And then it sets it out. Alright, so the only thing you have to watch is that you don't want to rely on the top lid because the top lid can spin. So this is a 4 mark one on this side, and then it's 3 on the other side. So if I rotate it, so the lid can move. So you want to look at the bottom, the edge where the media is. That's the plate. Okay, so we're going to put bacteria. We're going to do a simple streak plate, a 3-zone streak plate on a half plate. And so we'll start with E. coli. And we'll get it out of the incubator. E. coli. Alright, so it's going to be pretty quick because I can do the first zone on each plate with each bacteria. So I sterilize my loop, then I go in and I try to, I either have to go for an isolated colony, or if it's a pure culture plate, which these were from the original stocks, which are pure cultures, I don't have to be as picky. But where I do have single colonies, I want to go there. And so what I do is I will put a little bit light zone on each of the sides of the plate, and I can go back and forth because they're both sterile either way. And then I will flame it and then do zone 2. And so zone 2, I touch out in the area that is not the area that does not have bacteria. So then sterilize, and then go in here so I can cool the loop. So I come in a couple times and then come out. You have to have a light touch because the EMB and the McConkey agar don't have a lot of agar in them, so they can be a little bit sensitive to tearing. Go into this one, snake out, and so in the 3rd zone we snake out wide. And that one is done. And I need to, put what sample this is. This is number 1, E. coli. And the date is on there. Okay, and that is done. So I'll put this plate over here so I don't get confused. And then I just go on and streak the rest of these. And so I'll pick this up when we get the results and show you then. So I wanted to go over how I did the streak plate on the split plate. Here is the agar plate. And then I put the numbers on the back of the plate. So like number 1 sample was on the back, so I know where I'm going to start. I flame my loop, and then I go in and get my sample, and then I'm going to just smear it back and forth on the first inch of the plate, and I go wall to wall. Okay, then I flame again, and then I can go on and do the next one, and then I just go in a little bit, and then I stay out. Then I flame, and then I go in here a little bit, a couple times, and then I stay out and I start wandering. So you have 3 zones. Here's 1, here's 2, and 3. And you expect that by the 3rd zone you should have individual colonies showing up here. Okay, if you're doing the same, if you have split media, and you know this one was EMB on one side and this is MacConkey auger, you can speed up the procedure a little bit. So we'll get our bacteria and we'll streak the first zone back and forth, and then we can just go to the other side and streak it as well without flaming again and without getting new bacteria. Because they're the same bacteria, these both plates are sterile, and so I can do that. Then I can flame, and then I will twist my plate, and then I will go into the first zone a couple of times and then stay out. And then I can actually go over here because it is again the same bacteria. If it's different bacteria, then you have to start, you have to flame it and make sure it's sterile. But here we're the same bacteria and we're the same dosage. And so we go in a couple times and then we stay out. And then we're done with that. We flame it again, and then we twist our plate, and then we go in here a couple times, and then we wander. And then over here we don't have to flame it again because it's the same bacteria. Go in a couple times and then we'll wander. Okay, and then we'll flame it when we're done, and then this incubates at the appropriate temperature. And that's how you do a split plate, streak plate. If you do just a regular plate with one media, you do the same idea, but everything's larger. So you go in and flame and get your sample, and then you go back and forth in the first quarter of the plate and hit wall to wall. Then you flame, rotate your plate, come in here a couple times, and then just stay out. Always proceeding forward, flame, come in a couple times, and then each time that we flame, there's fewer and fewer bacteria brought out from the previous zone, and so then the sample will be less bacteria streaked. And as you pull the loop across the surface of the plate, bacteria drop off, and so more bacteria drops off at the beginning, and there's fewer left on the later zone. And then by the time you get to the end, if this is your fourth or your end zone, then you just start to go wide. And then you expect that the bacteria would be now pretty well separated, and you'd have these isolated colonies growing up. So in 24 hours, you used to see one bacteria turning into 10 million bacteria and forming a colony. And then you flame it one last time, and then you incubate this at appropriate temperature. Okay, that's it for now. We'll get the results next time of the real bacteria. Thanks.